I'm Elizabeth McNeil, and my friends here are joining me on the panel. While we wait, I'm going to just, I'm going to introduce myself. I'm a pediatric neurologist who was actually at FDA for 9 years before moving to NIH, and I think that one thing, I'm also looking for a place to plug this in. My little slides here. Bear with me for a moment. I'm going to ask the panel to introduce themselves as I get myself organized. Starting with you, Brenda.
Hi, I'm Brenda Wong. I'm a clinician at Cincinnati Children's Neuromuscular Clinic. Besides being a clinician, I've been involved in clinical trials for Duchenne. So I'll be sharing on the perspective of a clinician and a PI of Duchenne studies.
Hello, I'm Miranda. I'm a professor at the University of Western Australia, and I've been involved in research into skeletal muscle for about 30 years and very interested in developing therapies for Duchenne muscular dystrophy and muscular dystrophies, and I'm also involved with 2 committees on the Treat NMD network that are relevant to these kind of discussions.
My name is Yaacov Anziska. I direct the Muscular Dystrophy Clinic in Brooklyn, New York, and I'm particularly interested in the quality of life and pain and issues affecting boys with Duchenne muscular dystrophy, and also looking at particularly vulnerable populations, lower socioeconomic groups with Duchenne.
Hi, I'm John Krause. I'm from GlaxoSmithKline, the project physician leader for our exon 51 compound, GSK 2402968. And I work in a part of our company called the Neurosciences Medicines Development Center, where we focus on taking drugs through their late-stage development to registration and hopefully approval.
My name is Giles Campion. I'm Chief Medical Officer of a company in the Netherlands called Prosensa, and we've been working actually together with GlaxoSmithKline. They are responsible for the later phase development of the exon skipping program, and we have been involved in developing the earlier compounds. And I've been with the company about 2 years. Prior to that, been working in the pharmaceutical industry for about 20 years, and my original specialty was in rheumatology.
Thank you. Okay, we're going to go through a little bit about the stuff that happens before the clinical trial, the things that get the product towards the clinical trial. I'm going to give a brief overview, and then I'm going to rely on the panelists to give more insight from their particular areas of expertise. So the overview, somebody has an idea. After that idea, somebody has to explain the scientific rationale. We need to have chemistry, manufacturing, and compositions so that we know that this product can actually be made. And then we go to the preclinical, where we'll talk about proof of concept, mechanism of action, safety. And I should say that at least in the U.S., when I say safety, what we really are talking about is the toxicology to figure out whether it's safe to put into humans. It's not whether it's going to be safe in humans, it's whether it's going to be safe to put into humans. And what we looked at when I was there was what are the off-target or adverse events profile associated with this drug. We know that not every drug is going to be safe. Matter of fact, we presume that most drugs are going to have adverse events. But in the label, we like to be able to tell people sort of what they can expect as a potential adverse event from that drug so that people can determine whether those are adverse events that they are willing to accept for themselves or for their child. PK data also comes in preclinical because we get some PK data from the animals, as will be explained to you, and then there's PK data that actually comes during the clinical trial. The clinical trials, the more important things are, what are the outcome measures? In whom will you be testing, and when? Is there a therapeutic window that is going to be best to look at? Okay, so everything starts with an idea. You have some brilliant academic or someone in industry who says, I think that this product is perfect for Duchenne or Becker. So what they will need to explain is, well, why do you think that? Do you know something about the mechanism of action? Is this product supposed to be disease-correcting? Is it supposed to be disease-modifying? And if you think it's going to modify disease, which aspect of disease is it going to modify? That's important because that helps you later on pick out your clinical outcomes. Because if you don't know what it is your product is supposed to be trying to do, then you don't know how to quite gear your trial going forward. CMC, as I mentioned, you can have a great idea. It helps to be able to actually make the product. So therefore, you at some point are going to learn whether you can actually make the product or not. Preclinical, you have your various and sundry models. Probably don't have all the models there, but I've got dogs, cats, and monkeys, because non-human primates are something that we look at for some of the testing later on in FDA. And then, as all of you, or many of you, I'm sure know about the MDX mouse and the golden retriever. In preclinical, as I mentioned, at FDA, what we're looking at is, okay, well, how are you planning to deliver the agent and the appropriateness of the dose amount and frequency. If you have a huge dose of product that no child is going to be able to ingest, well, that's a problem. We need to figure out a different formulation. If you have to take the medicine every 2 hours, even the most dedicated parents are going to have problems adhering to that schedule. We need to figure something out. What level of exposure do you need to see the desired effect? What level do you need to see toxic effects? Because that's going to help in our calculations later on. Is there any off-target toxicity? What's the safety margin? If something goes in, usually it comes out. How does it come out? And does it affect the organ it comes out of? If it doesn't come out, are there any problems with it staying there? And then clinical trials. The who, what, where, what does the sponsor think this product is going to do? Again, that's going to help us get some idea about outcome measures. Whom do you select? Knowledge of toxicity, potential drug-drug interactions, expected benefits can all impact your inclusion and exclusion criterion. And you're going to hear more about that. How many people do you need in the study to show an effect? Is there any chance that, if you talk to the community, is there any chance that you can actually get that many patients? Because if I need, thousands and thousands of patients and the community tells me I can't get that, that's a problem with the trial, because that's part of the delay is you can't recruit to that trial. If you need 20,000 people and you can only get 1,000, you're going to have a long delay. And I'm sure that some of my folks in industry here will attest to that being a potential problem. Just nod your head then. Yes. Thank you. That was your cue. Nobody nodded. Anyway, when do you have to give the product? Some things, are you giving it prophylactically? Are you giving it to reverse disease? Are you giving it to modify or to stabilize disease? You do have to have some idea, because that's going to tell you which population, again, you're going to want to use. And different products may have different aspects of these. So you've already been hearing about the path forward and the different phases, so I'm not going to spend time with that. What I'm going to do, because I know there are probably still questions from the last session, which sort of fed into this session, is turn to Miranda, because you're my preclinical soul, and let her speak.
Okay, so one of the points that I think is interesting, particularly in the context of some of the earlier questions with the FDA being so busy, is actually sorting out which of these interventions are really worth going to clinical trials. So sometimes you get a research paper published, often in a very high-profile journal. People get very excited about it, and there's a lot of pressure on to proceed to clinical trials. So one of the things is I think you've really got to pause and check that those observations that were made, for example, in a mouse model, are perhaps validated independently by another group before they proceed. It gets very exciting. People say, let's go for it. I think you need to be very solid on your science before you think it's really worthwhile going to the clinical trial. And sometimes I think they progress too rapidly. So the work that I've been involved in from the preclinical point of view is, first of all, trying to get standard operating procedures in place so we can compare drug A with drug B and really assess the relative merits of that in a scientific context. So that's really important. So first, really sort out the science. The other thing that I'd like to make a comment on relates to a committee I'm on with the Treat NMD Clinical Trials Committee called TACT. And this has been set up really to check out the whole scenario of whether an observation that looks very exciting as a potential treatment for something like Duchenne muscular dystrophy really is likely to work in a clinical trial. So as I said, it can be very exciting, it sounds very sexy, very terrific. They will sit down with a wide group of people who are both basic scientists, parent groups, big pharma, regulatory bodies, and they will look at the entire picture. So they're an independent group. They're not going to allocate funding. They're merely going to look at the whole picture of this proposal and see whether it is in a position that it should go forward to a clinical trial. So the parent groups like Parent Project Muscular Dystrophy are really working closely with this TACT committee. So before someone comes and seeks funding, they will actually get their project assessed by the TACT committee, and they will make a comment on whether they think it's terrific and ready to go, or if something more needs to be done. In a way, they're a filter. So I think that's a very important step that has been set up, and it means as an independent group really checking it out before it gets caught up into this big pipeline, which is very demanding, takes a lot of time, a lot of resource, a lot of money, and I think that's a really good thing to have put in place.
As a full disclaimer, I actually sit on that committee, so this was not an advertisement, but I do participate in the committee as well. Was there anything that you would wish to add, Miranda, before I turn to the gentleman on my left?
No, I really just want to say I think that was not around before, and I think it avoids people rushing in on sort of fueled by hype, if you like, and presumably then if you can make a better case for therapies going forward, that will actually remove some of the noise from the system and perhaps streamline other ones going through it. There's often a huge vested interest by people to get their product or their drug into a clinical trial, and that just serves to pause for a while, which I think is good. So I would recommend that any parent, for example, asks if it has been put forward to a TACT review, and if it has, you can ask to see the full report for that if you wish, to see exactly what was said about it and so forth. So that information is available.
Thank you. Gentlemen? This is the industry folks.
Yeah.
We're gonna...
Yeah. Well, what I was asked to talk about was the decision-making process for a small company, a biotech company, taking a new product through to the IND stage. And what the IND process does is it's the opportunity for a company to present all the information it has on the compound so that the decision can be made by the FDA or whatever regulatory authority you're applying to allow clinical studies to start. Now, of course, the common questions that we get from patients and their families is, why does it take so long? And why is everything so expensive? And why do things fail? And I think that for patients, I think that the most visible part of the development process is the clinical trial, because that's when you actually have the opportunity to participate in the development of the drug. But there are a lot of other activities, some of which you've heard a lot about in the talks today, that go up to building that whole packet of information that needs to be assessed before the approval to go ahead with a clinical trial is taken. And I think one thing that tends to be pretty invisible is the huge amount of effort that goes into the actual chemistry of the drug. If you think about it, when you start off with the initial testing in animals, you're talking only about grams. And grams of agent are fairly easy to make, and they don't cost a lot. But when you're starting to think about the bigger testing you're doing, the preclinical testing, the long-term safety studies in animals, and then ultimately the human clinical trials, you have to take those grams of chemical and turn them into kilograms. And that can be very expensive, certainly in the case of oligonucleotides and some of the new therapies. That's a very expensive operation. The chemicals are expensive, very expensive to make. And not only do you have to ensure that you're making the right stuff, you have to be sure of the fact that once you make it, it's stable under conditions of storing the drug. You have to make sure that it's not affected by light. And in order to be able to have that assurance, you also have to develop a lot of technologies in terms of testing the drug. So there are a lot of activities that are ongoing just in terms of the chemistry of the drug itself. And clearly, as we've heard, you want to make sure that whatever's injected into your children is going to be safe there's not impurities, and that you feel confidence in it. Now, that's one element. Of course, what you have to do as well is then use that product, and it is tested in animals to get some sense of what the safety aspects of that drug is. And you start off with studies that are relatively short-lasting, and then you increase to look at a range of doses so that you can really understand what monitoring tools you're going to need in the clinic when you're conducting the clinical trials. You can understand from what happens in animals the type of testing you'll have to do to make sure that the product is safe in the individual. And you need a sequence of these things to happen. You need to work out how to upscale the manufacturing process so that you have enough drug to do your animal testing. Of course, in a biotech company, many companies have just been spun out of academic groups, and you start off with just a very few people. Then as your experiments become more encouraging and you get good results, so you have to go out and find finance. And as I said, manufacturing some of these compounds can be extremely expensive. So you have to work through the whole process of upscaling and manufacturing, doing your preclinical testing with the need to raise money. And what might be the quickest route in terms of a development path is complicated by the fact that you have to actually go out and raise money every now and again. Once you've gone through the preclinical stage and you've understood the safety, you've already heard about the fact you also need to know how you're going to deliver the drug. Are you going to give it orally? Is it going to be subcutaneous? Is it going to be intravenous? Then you have to go and make the final formulation of the drug one that is suitable to actually give to a patient. Once you've got to that sort of stage, the next stage where you're making decisions is where you're going to do your clinical trial and what sort of patient population you're going to test. The first clinical trial for which you seek approval, as you've heard, is generally purely looking at safety. However, it has been possible in some areas to actually not just do a single-dose trial, but do a multiple-dose trial. And in something like Duchenne, that saves you a lot of time because it allows you, with giving 4 or 5 doses, the ability to actually look at some sort of molecular readout. So is the drug not only safe, but is it doing the sort of thing you're looking for it to do? So to some extent, you choose where you do your clinical trials based on which authorities are going to allow you to do the trial that you think will give most information in the quickest way. Then sites, you decide about which sites, depending on where you think the patients are going to be. And particularly when you're working in a rare disease such as Duchenne, this is where it's incredibly important and useful to have patient registries that tell you, well, this particular mutation in the area that we're working, these are where the patients are to be found, so that you can try and minimize the amount of time that people take to travel to a particular site. Now, in the first study, the other thing you need to do, because generally you only look at a relatively small number of subjects, particularly in the dose finding part of what you're doing, is that you also need to be very strict about the inclusion and exclusion criteria, because you can't have too much variability if you're looking for particular effects, particularly if you're looking at molecular effects. So that means that you have to be very careful about exactly which patients you ask to get enrolled into the study. Once you've done your protocol, then you have to, and at the same time you're applying for approval either at the FDA to run the study or in Europe or wherever you're doing it, then you also have to set up relationships with the investigator sites. You have to have a contract. You have to write a consent form. You have to have ethics committee approval. And then you have to make arrangements for drug to be shipped to the site. None of that can go ahead until you've submitted all your information to the regulatory authority, and then generally there's a period in which they look at everything you've submitted. They may pose you questions, and then if you're lucky, they'll say yes, go ahead, which in the case of the FDA is about 30 days after you've submitted your claim.
Usually Friday afternoon at 5.
We got to be there, too.
So once you've done all that, you suddenly find going to the next part of the trial process is going to be very expensive, and that's when you turn to your big brother and ask him to take over.
So Giles gave a very good overview of what it takes to get the compound, the idea, to the clinical trial, and that applies to the later stage. So we'll focus for the purposes of this conversation on this idea of the so-called pivotal trial, that phase 3 trial. What does that actually mean? In general, when we use the word a pivotal trial, it is something that the regulatory bodies can look at and see if it gets at that question that was mentioned earlier. Is this truth or not? Are you able to answer the question? So going back from that, the most important thing when you're thinking about this pivotal trial, which is used by the regulators to make a decision on whether there is appropriate effectiveness of the compound, is asking the right question from the beginning. So the right question is the most important thing. And in the case of thinking about Duchenne muscular dystrophy, based on the mechanism of the drug we're working on, we anticipate to make a truncated, though functional dystrophin that should relieve symptoms of the disease. We also heard this morning that there is an endpoint that is generally agreed upon by regulatory bodies and experts that you can actually measure and use as clinically relevant, this being the 6-minute walk distance test. So here you have the ability to say, are we able to improve a specific symptom of, or a specific test in Duchenne muscular dystrophy that can be measured, that we can use as a proxy to say this thing will help with the disease and its symptoms. But the key thing is having that agreement with regulatory authorities, with experts in the field, that indeed the endpoint you've chosen to answer your question is actually clinically meaningful and will be able to interpret it at the end of the day. So the basis of the clinical study is the protocol. It basically says, here are the steps that are going to be done. And once you understand what your endpoint is, what your drug is, you take all the previous experience from the earlier safety trials, sometimes phase 2 trials with dose finding, and you begin to develop your population that you want to test. So I've heard some of you mention ambulant, non-ambulant studies. You know, Duchenne muscular dystrophy, the genetic defect is the same regardless of that stage of disease. But in doing the clinical trial, we actually start with the ambulant because we have the endpoint we can look at to begin for approval. As you get towards different populations, there's less certainty, so you need to do more exploratory work. So the phase 3 trial that we're running currently is looking at ambulant boys because we have an endpoint that we can measure that is agreed by regulatory authorities as being clinically meaningful. So at the end of the day, we can understand the data and make a decision. Now, once you have that protocol together, that's all well and good, but you need to have clinical investigators who are able to carry out the study. You need to have the patients and families willing to participate in the burden of the study. You heard the woman who's from Ottawa working with the GSK study. It is not an easy process. It takes a lot of commitment. And you need to decrease variability in the course of the trial. So we have a whole army of individuals that work with the investigators, that at times work with the families to try and monitor and oversee the quality of the trial, the quality of the endpoints, and to ensure that the trials are being done according to what's called good clinical practice. So I've spent a lot of time talking about the question, the efficacy part, but actually, I think incredibly important in any disease state that you're studying is ultimately at the end of the day, the FDA is going to make an assessment of a risk-benefit profile. And one of the parents spoke about the fact that, you know, this is a disease that over time will result in death. So it's a very serious illness. The risk-benefit, therefore, may be looked at differently than a disease like arthritis, for example. So the safety package that we need to develop is to support the benefit that we see as well. So we monitor incredibly closely during the course of the trial any evidence of any safety issues that come from the preclinical models that we talked about, that come from the early clinical, to ensure that as we progress, the treatment is not only effective, but actually has a safety profile such that it is worth giving for a chronic illness. That can be many, many years of treatment. And I guess the last point I'll make, and then there'll be an opportunity for any questions at the end of the session, is the other difference towards the end of the trial when you're working on that phase 3 study is regulatory agencies want longer-term safety data and efficacy data. So typically, we follow trials for the course of a year, but we also have extension studies at the end where patients can be continued to be treated until the drug is either registered or deemed not effective. So we can have patients on compounds for years before going into registration to better understand that safety and efficacy. At the end of all that, we put together a package for submission that summarizes the efficacy information or how well it works, summarizes the safety information or what sort of adverse events the drug may or may not cause. And you try and put that together to determine, is this drug effective? Is it safe? Is the relative balance of its effectiveness and safety such that, yes, this should be approved, this should be available to persons with the disease? So there's millions of parts in that, but that's kind of a short summary in terms of the phase 3 part of the trial.
And I should also point out the FDA's involvement comes in the beginning when you have that first, let me open my IND, and they get 30 days to review all the material that you've heard about thus far. And then, let's say that the product is said, it gets the go-ahead, and then the clinical trial phase starts. That's up to the sponsor because the sponsor gets to determine how they're going to do their trials, where they're going to do their trials, and all the things that you've just outlined that go into actually running a trial. And then the FDA, in the case of Duchenne's, would have 6 months because it's a rare, life-threatening disease without a currently available therapeutic option, to have 6 months to take that whole NDA package and review it. So FDA gets the month in the beginning, then the sponsor does what they need to do to accumulate the data they need, and then hands it to the FDA, and the FDA gets 6 months. So there's a lot of why is the FDA taking so long comes in, but the question is where are the places to sort of trim the package and expedite matters is something that also has to be considered. And before I go further, though, I'm going to turn to the clinicians. Brenda, ladies first, sorry.
Actually, a lot of what I would like to share had been covered by the previous section, so I'm just going to highlight a few areas with respect to protocols, outcomes, IRB, and hurdles and challenges. With regards to protocol design, I think we've heard how crucial it is to have the correct protocol design. Otherwise, the data that we accrue would not be valid for analysis, and that would be a waste of time, effort, and money. And with special attention to what we have learned from our Atulin trial, I would like to just share some aspects of it with regards to the logistics, as one parent had mentioned about the timing of doing tests. For example, we would like to assess the 6-minute walk and the time function test when the child is not fatigued. And so really, the logistics of conducting that trial, we have to ensure that the evaluations are done early on during the day and without the fatigue factor coming in and affecting results and validity. So I think that aspect of protocol design, it's really up to the local site for the investigator to work out with the research coordinator to work out the schedule for the patient to be studied. And I thought I'd just with regards to protocols, we usually talk of 2 keywords really to summarize efficacy and safety. and with regards to efficacy, we did talk a lot about primary outcome measures and secondary outcome measures. And again, I think knowing our patient population, there is a huge spectrum of variability. And hence, some parents may not quite understand why some kids are not eligible, even though they are ambulant. Because if we have a cohort that is so widely variable, as we have learned from our study with ataluren, that really throws in a lot of variability at the end of a year. And hence, you are going to affect the results, and the study will be underpowered. And so you would really want to ensure that the population that we're studying would be pretty homogeneous, so to speak. And so just discussing with your clinician and the investigator as to why certain cutoffs are made, like, for example, like the ability to get up from supine to standing in less than 7 seconds. So these are issues that families may want to bring up and discuss with your investigators. If you say, well, my child is ambulant and yet, we are not eligible. So just these little details so that it would really affect the outcomes for measuring efficacy to ensure that the study is conducted in the right manner. The other aspect, I think, is safety, which we have talked about safety with regards to FDA, but I think in terms of the clinician or the PI conducting the trials, it is the major responsibility of us to really ensure that the trial is conducted according to the monitoring for the safety of the drug. And so that aspect is really covered by the IRB approval. And so I know sometimes the IRB process may take long, but that's the ethics committee that really safeguards the rights of the human subject in clinical trials. So the IRB may not approve a study, and one would want to kind of talk to the site and understand what the concerns were. And I think IRBs are not out there to deny an approval, to disapprove drugs, but if they do have concerns, it's usually related to safety issues. And I think the hurdles and the challenges that we find comes a lot from the urgency of getting a drug that will work, and I think that really sometimes it's what Dr. Kraus has said, that you really have to be fully informed of the pros and cons of being in a study before you sign consent. So ultimately, the consent comes from the parent and the family and not from the investigator or the clinician pushing you to sign up for the study. And I think we've learned from our trial that, the boys themselves need to be consulted. I mean, they are the ones who's going to go through the trial. And there was a boy who says, I was not asked, and I don't want to do the trial. But when the parents discussed with him, he was open to it. And I think we must not forget the boy or the young man who is going to be the subject to be involved in this informed consent process as well. And I think with regards to competing trials, currently we are okay at this point, but with all the trials that's coming up for the next year, it definitely would be difficult for families. So do discuss with your clinicians, with your investigators of the site, to really gather all the information before you make the informed consent. Thank you.
Well, as a clinician, I just want to, reemphasize what Brenda said about safety. And I don't sort of want to throw cold water, on the process. But with respect to, apologies to my colleagues in the pharmaceutical industry, at best, a drug that's gone through phase 1, 2, and 3 trials, it's looked at a relatively small population over a relatively limited time. So as a clinician, I'm most concerned about, will this cause harm to the boy or young man with Duchenne? And I understand that people are desperate, and I've even had patients tell me that they'd be willing to, quote, roll the dice if there was a chance of really improving strength and life, even if there was a significant risk. So I feel my role as the clinician is to sort of have everybody take a breath, think, and then try to really make a very rational and informed decision about A, whether to participate in a trial, and B, if a drug eventually does come to market, is approved, whether to go forth with it or not. And I think if you look at, sort of the newspaper, it was in today's Times, for example, when we're talking about drugs that are approved for breast cancer, now the FDA may be taking that away, diabetes drugs, etc. You have all these drugs that have come to market, and we've only seen the side effects. Or in the case of somebody like Jesse Gelsinger, who passed away a few years ago in a drug trial for gene therapy, there are significant risks. So as a clinician, I have to say I'm more excited than ever with all the drugs in the pipeline that are coming. And I can now speak to patients and tell them that within the next few years, there will be drugs that are going to be tested in humans and hopefully will be approved for humans. But I just have to say to the parents and to the young men, with Duchenne, if a clinician is sort of waiting and wants to get a bit more information, it's not because he or she doesn't want, to help or doesn't want the medication to help. But we just have a duty. I mean, that's the sort of the Hippocratic Oath, is to make sure that no harm comes to the patient first. So again, I'm excited, with all the drugs that are coming there. But I just want to point that out again. That there are pros and cons, and just to really think through them.
And with that, we'll open the floor to questions. I see a gentleman raising his hand in the back. If you could just come to a microphone, sir. Thank you. Oh, there's a lady who precedes you. Ladies first, I'm sorry. Ma'am?
I've just been getting the impression that when looking at endpoints, that it's to make the disease better or to walk further with the 6-minute walk. In this disease, staying the same would be phenomenal.
Right.
My son's 19, and he has a great quality of life right now. He's independently going to college. If he would be joyous to sort of just, there was something that just could help the progression. So having as an endpoint that they're going to be doing more, better, faster, I don't know if that's a realistic endpoint, and maybe I'm wrong in my understanding of that.
I think, and I'll just say one sentence, and then I'll turn to the rest of the panel. But I think that different drugs can have different outcome measures for their protocol. So there might be drug A, for which they are trying to do stronger, better, faster, as you said, and there might be drug B that's its point is to modify the disease and stabilize it.
But I think it's also key in these clinical trials, they are placebo-controlled. And stability would be amazing. I agree with you. And as Dr. McDonald may comment, you see with placebo decline over time. And if you're able to actually stabilize over time, that can have important clinical effects. So that the trials should be able to detect that difference from placebo, which might be an increase depending on the drug, if it's disease modifying, for example, or may stabilize, or may show no difference. And being able to interpret that is the key thing to understand that potential benefit.
Yeah, that's absolutely right. All the recent trials really have been powered on the basis of stabilization of the disease, relative to the natural history, relative to a placebo-treated arm, which is expected to decline. I think it's also important to keep in mind that an endpoint such as the 6-minute walk test is really just an indicator of disease progression. And when you look at the history of the corticosteroid trials, we all know what impact corticosteroids have had on Duchenne dystrophy over the decades. Those trials initially came out in the late 80s, and really because of side effects and maybe modest efficacy at first, corticosteroids were really not clinically adapted or clinically employed by the medical community, and it's really been fairly recently that as we have followed these boys longer term and young men, we have seen the additional benefits in terms of pulmonary function, prevention of spine deformity, prolongation of need for ventilation, improved survival, improved arm function, and so forth. So I think it's important to note that when we make these decisions about this risk-benefit ratio or comparing efficacy with safety, really some of the efficacy may not really be known or emerge for many years after that 12-month clinical trial. But most clinical trials really are designed to take place over a shorter duration of time.
I think, that really highlights the need for the control group, because without the control group, you cannot compare the treatment effect that it is indeed slowing disease progression. And I think the buzzword is like clinically meaningful outcomes. And what is clinically meaningful, really, for those of us who are not familiar with Duchenne's, we will not appreciate it. Like for the parent who's got 18-year-old who's non-ambulatory, but, arm function is crucial, right? So to be able to give you a hug is clinically very meaningful. It's life-altering change. So I think what we would like, and been working on outcome measures, we really appreciate the family's input to say, for a non-ambulatory patient, what matters? Like for our patient who is able to lift up his urinal after a drug intervention, that is clinically meaningful. And so I think getting input from families will help us really work out for the group that we don't have like a 6-minute walk for the ambulatory group. And that's why, there's still a lot to be done. But that emphasizes the need for a placebo arm.
And also, I look at quality of life, and we've actually seen that quality of life does not necessarily correlate with strength. And I think what Brenda says, for example, if you could, I have a patient who's, wheelchair bound, who can barely move his hand, but he can draw, and he's an excellent artist. And to him, that's all he wants. So I think, yes, I understand the 6-minute walk test. Obviously, the biggest limitation is it doesn't help non-ambulance people at all. But if there was a way to measure that, and maybe looking at quality of life as an outcome measure, because I think everybody approaches their weakness in a different way. And I think, yes, we want to look at pulmonary function tests, et cetera, but I think there's more to it than that. So I don't know if there's a way to incorporate that for future trials, but I think it's not a bad idea.
Just a quick point on that is the primary endpoint is really what drives the size of the trial and discussions with regulators, but the studies have significant numbers of secondary endpoints. And I agree, the quality of life issues and the ability to capture individual achievement beyond the defined endpoints is important. How that will relate ultimately to regulatory approval is a different issue. But
it depends. I think that there's room for the community to give input, especially with the patient-reported outcomes guidance, as to what type of things are important for the community. But that poor gentleman's been waiting, so I'm going to stop talking.
Yeah, I just want to make a couple of points. Miranda, nice to see you again. I haven't seen her for ages. But, to suggest, I think, that there are hundreds of researchers over the last 20 years desperate to start clinical trials on flimsy mouse data, I think, stretching it a bit. I really wish it were true. In fact, the reverse is true, that, over the last 20 years, everyone's been, doing a wonderful job at curing mice, but have got blocked by this terrible pipeline that you guys have took, this human trial blockage that we've had. The second point I just wanted to bring up was about pivotal trials, the stage kind of 3 trials. And I think we need to understand that, that those trials are usually large patient cohorts traditionally. And they are double-blind placebo trials, and that's the traditional format. And personally, I think that's excellent. In certain circumstances, that can be very important to do those types of trials. But in Duchenne, I think, again, we've got to look at it differently. Why? Because we've got very low patient numbers in Duchenne. And and when you start to, as somebody said earlier, when you start to do exon skipping trials and you start to, look at kids who are on steroids, not on steroids, etc., you start to reduce a very small patient number into a very small subgroup numbers, and it becomes very, very difficult to get those, large numbers for clinical trials. Also, to go back to Craig's data earlier on, he showed very high variability in the natural history of Duchenne patients. And if you looked at his graphs, I thought they're really, really interesting. But there's a huge scatter in those graphs. And so, if you select a group of kids, for a trial, you could be picking kids of very, very high, very, very different responses to the onset of Duchenne, etc. And also, and I think there's another big increasing problem that's not being discussed very much, and that is a lot of families, and certainly in the UK I know, are starting to use, they're on prescribed medications, steroids, heart drugs and so on, and some families are already starting to use other unprescribed medicines for their children and not declaring them. And I think so in that way there's another huge variability within the Duchenne population that makes that stage 3, final seminal trial very, very difficult. And as you're from GSK, I'm sure you know only too well that a lot of the trials that GSK and other big biopharmas have done over the years have rarely actually ever given you that sort of like final data to say, yeah, we've got a blockbuster drug, we can now go with it. So I think for all those reasons, we have to look at a completely different way of looking at clinical trials for Duchenne. And I would, again, try and point out, I think, for us families, what we want to see happening is delivering a personalized medicine to a Duchenne patient. That would be a massive step forward from where we are at the moment. And a last point I just want to make is on this whole risk-benefit argument, because I hear whenever we have these meetings about clinical trials and about causing harm, the spectre of killing a patient is always raised, and that's that can happen. We know that can happen, and no one wants to cause harm to anybody. But does that argument actually take us forward, or is it just paralyzing us? If every time we have this discussion, we say, well, we've got to be careful, we can't do this because we might kill somebody. Well, yes, you might do. But you've got to think of the risk-benefit equation here. You've got to point out the risks to us and tell us exactly what they are so we can understand them. Unlike with steroids, when I go to see my clinician, he says, yeah, you can put my son Saul's been on steroids since he was 4. Here are the risks, Nick. But actually, here's how we're going to manage those risks and make sure that we can manage them. But just at the final point, but the benefits are a couple of years extra walking. Now, the benefits for Duchenne, you have to measure against the fact that that kids with Duchenne muscular dystrophy are going to die if you do nothing.
I think one thing that sort of perhaps hasn't been fully discussed is the fact the risk-benefit, part of the way you find out the risks is through the clinical trial. And unfortunately, that means during the clinical trial, the people who are in the clinical trial have the adverse events, and then you know what risks to tell people to anticipate. So that's how we learn about the adverse events during the clinical trial itself. And the decision on approval is based on, well, do those risks and benefits, are they reasonable for this community? And every disease has different risk-benefit equations. So it's going to be determined by the drug and the disease looked at together.
I'd just like to make one point. I mean, as a preclinical researcher, we're very keen to identify what might be the best point of intervention or the best target. And by the same token, I think it's very important to be very critical so that really the most appropriate ones go forward into this pipeline. And that's really why I'm saying that if someone finds a very good observation in a group of mice, it should be validated independently before it goes ahead. There's certainly evidence that it's often not in the enthusiasm. So I think it's better to pause and be just a little bit slow and consolidate those positive data before you proceed, because that's essentially what's going to represent the benefits in a clinical situation. So you want to be very confident that those benefits are indeed going to be there before you embark on the clinical trial. So just in response to that idea that there's not a lot of people wanting to set up things, I think there's a lot of people who want to propose clinical trials with a lot of interventions at this stage. So you can't do them all, so you've got to be careful which ones you prioritize.
Could I just make one comment to the point about the individual patient type outcomes? I think the advantage in terms of certainly the environment we're working with exon skipping of doing reasonably large trials with the lead compound is that it affects the largest number of patients. And I think the hope is that once we have defined that risk-benefit in a reasonably large number of patients, then we will be able to be much more efficient when it comes down to rare exons. So I think certainly the feeling is that we do this one right, then we will have opportunities to make efficiencies further down the line.
I just wanted to quickly address the point about patients using drugs off-label or using non-approved drugs at all. And I've definitely seen that in my practice. And again, some of this comes back to the safety issue. If you're taking non-approved drugs, supplements, for example, you have no idea what you're putting into your body. Or, what the drug, the nutritional supplement that says it is really is what it is. And taking even approved drugs, but for a non-approved purpose, I think can be risky as well. And I think all clinicians, we do prescribe off-label, for example, but again, there should be some evidence behind it. There should be some hints that there's efficacy behind this. So, usually when I speak to patients, I try to tell them, again, we discuss risk benefits, but, I do tell them that if there's really no evidence at all for it, I wouldn't recommend spending the money or even risking any kind of potential injury by taking these supplements or drugs.
Okay, I just have a quick comment. I'll be very brief. I'm Sharon Hesterly. I'm the research director at Parent Project. And since we're talking in this session about decision-making and how you take a drug to clinical trial, how you make that decision, I just want to introduce the concept of an opportunity cost, because it's something that we face all the time. We have limited resources. We don't have infinite funding, and we don't have an infinite number of patients to participate in clinical trials. So we always have this decision-making process. If we take drug A to trial, then we are making a decision that we're not going to take drug B or drug C, that it's not that everything goes. So a lot of times when people ask, well, why can't you just test something? And so it always starts with just. Why can't you just? But a lot of times the answer is we would like to, but we have to make decisions and take the best drug to trial. And we want to be very careful that we use our resources, especially as these things get into phase 2, phase 3. You're putting millions and millions of dollars. That's the point where you've got bigger companies like GSK, dedicating $400 and $500 million to bringing these things to trial. If you bring the wrong drug to trial, it means the right one may not go because you don't have the resources. So that's part of the decision-making process, I think, from our standpoint, that we try and be extremely careful to make sure we're putting the right drugs forward.
As a person who helped develop the 6-minute walk test and doing clinical trials, it seems to me one of the things we have to really think about is when we're doing these outcome measurements, are they sensitive enough to measure the change? And that's why we're picking these certain ages, because we know they're sensitive enough to measure the change. We're very much interested in quality of life measures, but if it's not sensitive to see a change, this doesn't work in a clinical trial. So that's kind of what we're trying to do in this natural history study, is look at a lot of different measures and seeing how much they change over time. And just by seeing if they changed over a 2-year period of time, if something, could you tell a difference between a 4-year-old and a 6-year-old or a 6-year-old and an 8-year-old on these clinical measures? Then you might be able to use them in a drug trial, because you could say they changed, or a 14-year-old to an 18-year-old, because we know there's this absolute regression of the disease. So that's one of the things we need to learn. So I think even if we're not having everybody in a drug trial, if we learn and do more to do registries to find out more about the natural history of what endpoints will work, it could work for everyone. The other thing is I think when you're in a drug trial, if you put your child, I hate to say this, you can't expect it to work. It's kind of, again, it's looking at the benefit of the individual versus the benefit of the society. And the drug trial is to find out, does it help for the society? And if the society benefits and we find out the answer, it benefits everyone. So I think really, especially when we're talking about multiple drugs coming down the line, we want to work more like the oncologists do. And really, we want to get more and more people involved in these drug trials. And then have the individual having the parents decide and the child decide which trials they want to be in, or if they don't want to be in any at all, be in a natural history trial to learn more about the disease. So we are looking at more at quality of life. We're doing quality of life measures on all our natural history trials. We've worked with PTC very extensively on the 6-minute walk test, and we have another problem in Duchenne muscular dystrophy. We've got this whole maturation thing that's going on at the same time as we're getting degeneration. So that these boys are growing, so from the ages of 4 to 7 or 4 to 8, they're growing and getting better. So then, if you're looking at a group from 4 to 7 or 4 to 8, you're going to have to show an improvement with a drug. But from 8 to 11, you might say it's got to level off or not show a decrease. And we have to look at different ways. How do they compare to controls or other ways to see if these drugs really do work? Like I said, in PTC, staying the same or even not declining as much was an improvement. So what we're saying is if the placebo group declined by 30% and the PTC group declined maybe by only 15%, and it was statistically significant, that would have been an improvement. So that would have worked for the 6-minute walk distance.
Just a comment on that in terms of the natural history. There's another audience for these data, aside from the regulators, the clinicians, and the families that are insurers, payers. That can vary from region to region, country to country. But what the natural history data we hope can do, and you alluded to some of this in the presentations, is changes that are measurable over the course of a clinical trial. Are they predictive of longer-term outcomes that are clearly clinically meaningful? Mechanical ventilation, cardiomyopathy, things of that nature that are clearly clinically relevant. So can you make those correlations? And those sort of data will be very much supportive in terms of our being able to discuss a test like the 6-minute walk distance test, which these insurers may not be very familiar with, and try and correlate that with other outcomes.
Hi. Yeah, I totally agree with what Ted just said. And I wanted to comment in terms of the outcome measures. And I just think the data that you guys are collecting is so wonderful because I'm a physical therapist, right? So I think that if we're looking at, it addresses the question that the gentleman asked previously about his IEP for his child, and that they're being pressured to put on goals that will improve. And I think that the same measures and the same paradigm needs to be applied for the results of physical therapy or any other clinical interventions, whereas if you can specifically change the rate of progression or the percent of change so that it slows the rate of progression or there's a different percentile, that should be considered success. And so I would just love for this whole group to speak with a united voice when it comes to PT from my standpoint, but any clinical interventions, if it's changing the rate of progression, that should be considered a benefit. And so the more we all talk that way and have a different paradigm, I think it's really good. Sir?
I would like to say something about all the process. And indeed, it can be speeded up, probably working on the bureaucratic aspects. So instead of waiting for 24 months to look at all the documentation, something can be done to speed up the process. I don't think that can be done something to avoid some of the proof of concept that are requested to move the process on. I'm a pharmacist, so I really believe that everything is requested by the EMEA or the FDA is strictly necessary for kids to be safe. And I don't want my son to enter in a trial if it's not safe, and I don't want to change because it's good as it is. So the only thing I really try to change is the bureaucratic aspect of the process that can be really, probably with money, can be more successful. And the other thing I want to say is that probably if I was able to go back in time 10 or 20 years, the only thing I would change in our community is the use of the registries, because we still now miss the bird eye view, the panorama view of this disease. And I speak on behalf of Italian families. We've been involved in naive trials for years and years and years, and our clinicians have been not collecting that data for a long time, and not for all the trial they started. So this is really something we really miss. If we spent 20 years collecting data, probably today we could do better than we are. So this is the only thing I would really like to change. That this is the reason why I invite everyone to collaborate more, doctors, clinicians, and families, to the registry, because it's the only real tool we have that can change everything we know about this disease.
Could I just make a comment in terms of the bureaucratic element of it? I think certainly in drug development, seen from our perspective, there's an awful lot of white space. And the white space is not necessarily in areas where things take as long as they take, but they're more in the times when document sits on people's tables waiting for them to look at them or to be reviewed, waiting to go through ethics, waiting for contracts to be done in hospitals, waiting for insurance certificates to be given. I mean, as you may be aware by now, there's 100. There are many, many different links to this chain in terms of trying to put a whole development together. Elizabeth Room once said, which I thought was a nice analogy, society is organized, certainly as far as traffic is concerned, with having various speed bumps and speed signs to make sure that we all do things in an organized manner. But if there's an emergency, and I would consider Duchenne as an emergency, you put on the blue light and you go through all those speed bumps because it's urgent that you get a new drug for this condition approved. And I think if we could start to develop a system where dossiers and applications for ethics committees, applications for regulatory approval had blue lights on them, I think it could shave an awful lot of time off. So I think to some extent it's society's approach. I think we could do a lot, and collectively in this room, something could be done to really speed up a lot of this white space that occurs in development.
I just wanted to emphasize that one of the things that we can control as parents is we can participate in clinical trials. And that includes natural history studies that have no chance of directly helping us today. But you cannot test these drugs without measures. And there are ongoing outcome measure studies right now that are not fully enrolled because there aren't enough participants. So if you want there to be an outcome, participate.
I have just one small point, and it may sound like I'm saying make haste slowly, but speeding up the process does not always mean making a shorter trial. That if you have a trial that's too short to be conclusive, 6 months is not enough to see a difference. You have to decide whether having an extra visit and another 12 weeks or enrolling an extra 10 patients, if your sample size calculation is off, if your standard deviations disperse over time, what amount of money will it take to do that extra visit, those extra 10 kids, versus the loss of the entire study? So, yes, we need to move as fast as we can, but we need to do our studies like we mean it and not try to cut costs by minimizing the number of patients or the duration of the study.
I'm getting the look of perhaps my time has run out, but are there any more questions before?
I just want to encourage everyone here that knows other Duchenne families that aren't active to register. We've got to get the trials populated. And I mean, I think there's a lot more patients in this country than what we've got in the registries. So everybody needs to encourage other families that they know to register.
A question. As a researcher in another field, are there standardized protocols that are blanketly approved by FDA? Why not? As a taxpayer of the United States, why can I not ask for that? My biggest disappointment was I had to drive by Washington, D.C. to get here. My second biggest disappointment is that I will have to drive back by it to go home. Why can we not have these blanket approvals that a 6-minute walk is a standardized, approved, measured outcome that doesn't have to be rationalized by every institutional review board in the United States at every university that's doing research? That it isn't accepted by FDA, accepted by University of Cincinnati, University of Florida, whoever it might be, that can cut through the red tape of having to deal with that. As a taxpayer who pays for FDA to function and NIH to function, I should be able to expect that they can make those relevant decisions as professionals that I have to make as a professional in my field, and do some of that to help facilitate the small biotech company and the larger biotech company to put the blue lights on to go through those speed bumps.
As an ex-FDA employee and a representative of your tax dollars, thank you very much. My payday's on Friday. I would like to say that actually when someone comes in with a standardized measure, we do accept it. That's not the problem. I don't think that anybody said to anybody, no, we aren't going to accept the 6-minute walk test. Unless
it also depends on the maturity of research in the disease area.
Right.
So if there haven't been any treatments for a disease, no matter what it is, there's typically a lack of a uniform outcome measure that might measure an effect. So that's where it gets a bit difficult, but for established diseases and for established endpoints, 6-minute walk distance has been used in other diseases. That's been less of an issue in talking with regulators. You have some understanding of that. There are a variety of other issues that come up, but in general, one of the reasons for us using the 6-minute walk distance test is it had a regulatory approval in the past for other diseases to make it easier to go forward. That doesn't mean that the endpoint is perfect, and this is also why we have other endpoints in the study. As we learn more and more with natural history data, with the data from our own trials, there may be better endpoints or biomarkers that will be used. Then that will be a new discussion with the agency because that will be a novel measure or endpoint.
But I should also point out that the biomarker qualification team at the FDA invites people to come forth and propose biomarkers to be qualified for context of use in any disease. So therefore, there is an attempt, but the thing is, people have to come to the agencies with the data. The agencies can't go out generally and say, do this, because we can't do that. Sir.
I just want to double down on that point. There are numerous tests that are employed across the industry for various and sundry diseases, either at the clinical level or at the preclinical testing level, which we all know what they are, and we know that they are the gatekeepers. We just do them. We know we have to do them. It's expected. You don't go and ask the question. So so you can safely drive by Washington. It's okay. We're not reinventing the wheel each time unless, as you point out, a sponsor just insists upon reinventing the wheel, which is not FDA's doing.
There's nothing we can do.
So the difficulty in the case of the 6-minute walk is that no one knew until relatively recently whether or not this was a useful measure at all. It's useful it started off in cardiovascular disease, which is a fundamentally different problem of cardiac and respiratory insufficiency. It has little to do with muscle wasting, at least at the earlier stages of the disease, in contrast to DMD, which is muscle wasting very early on, and to the contrary, cardiovascular and respiratory problems later.
Democratic issues. I think there could be some discussion, though, about the IRB and ethics committees.
that have to be done on every single institution. And as a community, we could ask, could that process be done in a better way? Because that is a very significant barrier, and it takes a long time. So that's one, I think, as a community, that's where you could make a real difference. Other things that take a long time, I know when we do our trials, is contracting. Contracting is a mess. It's not easy. Some companies are easier for the University of California. Some of them we have blanket contracts with, but others of them will take 6 months or a year just to get to do a trial. So these are things that slow down trials.
Which are not government, and so therefore...
Which is not yes. Right, that's not the government. I mean, that's...
And as we've alluded to with a rare disease, in order initially for these earlier studies to be able to power them, you're going to dozens of countries throughout the world which have their own regulatory processes, ethics process. So it's very difficult to standardize that globally.
Sir.
Just to give some perspective on how rapidly things actually work, we were approached by PTC to develop and validate the 6-minute walk test in Duchenne dystrophy in 2007. And literally the 38-center, multi-center clinical trial literally started in 2008 before we even had our 1-year observational natural history data on the 6-minute walk test. And, I can't tell you, we had a tremendous amount of convincing to do, even in the Duchenne research community, as to whether the 6-minute walk test could be safely performed in the Duchenne population. We were, arguing with the Europeans. We were arguing with the physical therapists. We really had to demonstrate this in Duchenne. I think at least this is an example of one endpoint that seems to be working in Duchenne dystrophy, and the PTC trial, it ended up being actually the least variable, the most sensitive endpoint. We're hopeful about other endpoints. We don't have validated biomarkers right now. We're hopeful that there will be future studies in the very near future to actually compare potential candidate serum biomarkers with some of these clinical endpoints. I think that's going to be a huge benefit. We're also hopeful that we can develop some very innovative measures of upper extremity function as well.
I would just like to encourage folks here that, I know it seems a long time, but just the example of PTC therapeutics. I believe phase 1 was in 2005, and within a year, like 2006, we had the phase 2A. And then you worked on the 6-minute walk as a primary outcome measure, and the phase 3 trial was started in 2008. So that really showed that things can move fast on that timeline. And I think that's encouraging to know that that has happened. And I'm sure, with the lessons that we've learned from that trial, we can improve on our timelines. So I'm hopeful.
And the one thing I did want to say is that NIH does, as I was just quickly reminded, they've just set up a new call to do NIH, the new NIH network, Next Network. They're asking for 25 centers around the country to come together to perform neurological trials. In this trial, they're asking for a standardized IRB, one yes. IRB. So they are really looking to help the, to increase the translational activities. And so they're very familiar with, they're trying to understand the problem. They're looking at new ways of going about it. They're trying to set that one up as a model, and they're going to see if it works.
And we're accepting applications for that starting late fall, early spring for NeuroNext. Our website, sorry, this is a little advertisement because I am the scientific director for NeuroNext, so I want to take advantage of the moment. www.ninds.nih.gov/NeuroNext. We're waiting for your applications, yours. So I know that I'm over time, and I'm seeing Pat's giving me the mommy look, though.
No, are there other questions? I think it's never going to be close enough, but at least, the good news is we're talking about clinical trials, right? A couple of years ago, we weren't even talking about clinical trials. So now that we are talking about them, to find ways to streamline and work together to not go around the safety issues or around the IRB issues, but to streamline them and cut off the white space. And I think that's really what we have to focus on as an advocacy community. How can we help all of you cut down on the white space so that we get to those trials and hopefully get to therapies for our children. So thank you very much. I hope the streaming went well, and I appreciate all you do. Thank you.
Thank you all.